Hello, everyone. Thank you all so much for joining today. We're going to begin the webinar promptly at noon Eastern time, so in 5 minutes. Hi, everyone. Thanks for joining us today. We're going to begin promptly at noon Eastern time. All right, we're going to go ahead and get started. So hello, everyone. Thank you all again for joining today. Welcome to the NOAA Omics Seminar Series. I am your host, Catherine Egan. I am the NOAA OAR Omics Coordinator, and I sit in NOAA Ocean Exploration. I also sit on the NOAA Omics Working Group as a subcommittee co-lead. Omics, which is a suite of tools used to analyze DNA, RNA, proteins, and metabolites, has become a large priority here at NOAA in the last few years. We established this seminar series in an effort to increase transparency and collaboration, and also highlight the incredible omics-related research currently underway, both within and outside of NOAA. The seminars from this series are all made available on YouTube and posted up on our Omics website. I will drop a link to the website in the chat in just a moment so you can view our past seminars. As participants, you all are muted, but feel free to type questions into the questions for staff box throughout the presentation. At the end of today's presentation, I will read the questions for our presenter to answer. So with that, our presentation today is titled Open Ocean to Octocorals, Oh My, Applying Omics Tools to the Exploration of Remote Habitats and Species, presented by Dr. Meredith Everett. So Meredith, while I go ahead and introduce you, I'm going to switch over and give you screen sharing permissions. Okay, so while Meredith gets set up, I'll introduce her. So Meredith holds a PhD in marine biology from the Rosenthal School of Marine and Atmospheric Science, or RASMS, at the University of Miami. Applying genetic and genomic tools, she has worked on marine organisms ranging from teeny eukaryotes and phytoplankton to salmon to corals. Her current research focuses on using genetic and genomic tools and techniques to study deep sea coral and sponge communities to better understand their composition and distribution, as well as their importance as habitat to fish and other invertebrates. As part of this work, Meredith has been developing environmental DNA, or eDNA, approaches to understand the biodiversity in deep sea communities, and applying these methods to communities across the Pacific Ocean. When not in the lab or out at sea, Meredith can be found cycling and rock climbing around Washington State, or just hanging out in the garden with her family at home near Puget Sound. So Meredith, thank you so much for presenting today. I will now pass it off to you. We can also see your screen very well.
Thank you, Catherine, and thank you for everyone attending today. So as Catherine said, today I'm going to talk to you about Open Ocean to Octocorals, Oh My, applying omics tools to the exploration of remote habitats and species. So first up, what do we mean when we say ocean exploration? So in the context that I'm going to be talking about, what we mean is that ocean exploration can provide a multidisciplinary first look at an unknown or poorly understood area of the seafloor and water column, and an initial assessment of an area's physical, chemical, geological, and biological characteristics. And so today I'm going to talk to you about how we can use omics tools for ocean exploration and examine kind of three different quote unquote types of exploration: an initial exploration of completely unknown areas of the seafloor, or exploration as a tool for characterizing individual sites for better management and management decisions, and then exploration in the context of understanding poorly understood animals, such as deep sea corals, biology better, how they connect to one another throughout habitats, and how we can better understand what species are present in a habitat. So one of the challenges of working in the deep sea is that it's just a really remote and difficult to access area for study. So areas we work in are often geographically very remote. So for instance, one area that I'm going to be doing some collaborative work on in the coming year is the Pacific Remote Islands Marine National Monument, which is way out in the Central Pacific. So it's just physically difficult to get to. But this can also be true of coastal habitats when we're talking about accessing the deep sea. It requires a lot of specialized technology to operate at deep ocean depths, and that can really limit our ability to really explore these habitats in depth and collect samples from them. And so as a result, we often know very little about what species live in these habitats or much about their basic biology and connectivity. But despite the fact that we might not know a lot about them, communities in the deep sea are subject to an increasing number of anthropogenic challenges. These can range from fishing, where they can be subject to bycatch. They're increasingly subject to climate change pressure, even though when we think about the deep ocean as being kind of this remote, cold habitat, it's dependent on the surface ocean for all of the food that trickles down as marine snow into these habitats. And so climate change impacts in the surface ocean can eventually make it to the deep ocean. And then we have more novel challenges that are starting to really become apparent, particularly things like deep sea mineral mining. And some of the areas where mining leases have been proposed, no exploration has been done. And so we don't really know what, if anything, is living in some of these habitats. And so going forward, what we want to do is use a variety of tools to better understand the pressures that are getting put on these deep habitats. And that in turn will allow us to respond in ways for conservation, restoration, and minimizing the impacts we can have across deep sea communities. So surveys in the deep sea require specialized technology to access them, and there aren't that many dedicated programs for exploration. So when we carry out exploration, we really have this need to maximize this number of samples we can collect and what we can do with those samples and surveys. So what you're seeing now is images from NOAA's Ocean Exploration Program, which is the only NOAA program that's dedicated exclusively to exploration. And in the deep sea, understanding the biology in particular typically involves the use of remotely operated vehicles, or ROVs, like the Deep Discoverer, which is pictured here in the upper right. And these can collect video and imagery of the seafloor in addition to taking samples. But in addition to these more traditional technologies, we're increasingly applying new techniques such as environmental DNA sequencing, which is something that I'm going to be talking a lot about today, to complement these traditional survey methods and allow us to better understand the biology and biodiversity of seafloor communities. So we can use omics tools to address exploration challenges such as the limited field of view of the ROVs, the limited sample capacity for taking individual samples, and use it to enhance our ability to identify biodiversity and populations at risk. By characterizing the overall biodiversity in habitats, we can apply omics tools to study connectivity amongst different populations of species, and we can also apply omics tools to better understand species and their local evolution in the deep sea. Now, today I'm going to be focusing primarily on these first 2 points, characterizing the overall biodiversity of deep sea communities and the applications of omics tools to studying connectivity within populations of a specific species. So one way we can explore use omics tools to explore the biodiversity of deep sea habitats is through the application of what's called environmental DNA sequencing. So eDNA is simply DNA that, rather than being extracted directly from a tissue sample, is extracted from an environmental sample such as a water sample, where we filter the water, collect things like the cellular material that are being sloughed off from the animals in the water, and we're able to collect that on a filter and sequence that directly rather than having to sample every single animal in a habitat. This tool is becoming increasingly popular for understanding baseline biological inventories. It can also be used to detect rare species, species like invasive species, and to better understand things like trophic interactions. And so in the deep sea, we're now using environmental DNA as a way to complement existing operations such as visual surveys and get more comprehensive information about the habitats. So I'm going to be talking about applying environmental DNA sequencing for 2 types of exploration. Initial kind of first contact exploration of completely unknown habitats and locations. So this is very much a first snapshot of data from a specific environment, but also how we can apply these tools as part of more comprehensive survey efforts. For example, for doing surveys in essential fish habitat zones on the West Coast. Now, the sample collection process for each of these is actually very similar, but where it will vary is in the details, such as the depths covered, the number of replicants you take, etc. And one good way to think about environmental DNA is that it's just a different type of net. So just like if we were going to be using nets for doing a traditional ecological survey, and nets can have different mesh sizes, we can think of eDNA as having different quote-unquote mesh sizes as well. And that corresponds to different levels of sampling specificity. So on the one hand, we can use environmental DNA to detect a single type of animal at very, very high sensitivity. So we can detect really rare things very sensitively. We would use a technique such as quantitative PCR or digital digest PCR to do this. But on the other hand, we can use things like amplicon sequencing to detect all of the metazoan or all of the bacterial components of a whole community. However, at this farther extreme, what you lose is specificity. You often at that level can only detect down to the family or genera level as opposed to individual species. So for what I'm going to be talking about to you today, we're going to use this really broad sampling to get that first contact look at an ecosystem. But we're also going to apply kind of a middle ground sampling strategy where we want to be able to detect many species but still be as specific as possible. And so when we look at things like octocoral surveys, we're going to pick a set of molecular markers that allow us to maximize the number of species we detect but be as specific as possible, kind of right in the middle between the two extremes. So I just want to walk you through what the environmental DNA sampling process looks like as we carry it out. And this is regardless of the type of survey we're doing. So the very first step, of course, is to collect water in the environment. You can see here in the upper left, we've got a picture of a CTD rosette. That is one technique we often use to collect water. You can also use ROVs or AUVs to collect individual samples as well. For the work that I do, we're typically collecting 1 to 2 liters of water at a time. We bring these back onto the ship where we have a filter rig set up, and we filter the water samples we've taken on the ship, and then we preserve them in a DNA buffer that preserves the DNA at room temperature for transport back to our lab. This is really convenient and important because it means we don't have to worry about carrying a lot of ice or freezing samples on the ship. And if we're out for a month at a time, the samples are stable until we can get them back to the lab for extraction. Once they come back to the lab, we carry out an old school style extraction called a phenylchloroform DNA extraction on each of the samples. This produces really, really high quality DNA, which is really important because DNA can often be degraded in environmental samples. And we elute in about 50 microliters. So out of 2 liters of water, you end up with 50 microliters of DNA to work with. I primarily use a process called amplicon sequencing on an Illumina sequencer. This produces millions and millions of sequencing reads from my targets of interest. In the lab, I then run it through a bioinformatics pipeline that sorts out the different sequences into different types and allows me to get species assignments. And then I can use these species assignments in downstream analysis, looking at the ecology, pairing these assignments with what we have observed in video surveys, and just carry these downstream through the process of exploring the habitat. So to get a little more in depth on our laboratory and bioinformatics methods for both of the different types of exploration I'm talking about today, for PCR amplicon sequencing, we're using 18S libraries that target all the metazoa on the water column for our first contact exploration. And then we're using a mute-S, which is an octocoral specific marker to look at octocoral community diversity on the West Coast. And on the West Coast, we're also using a 16S marker to look at the fish community that's associated with deep sea coral communities. These sequencing libraries are sequenced on an in-house Illumina MySeq sequencer at Northwest Fisheries Science Center. This produces millions of reads per run, depending on the chemistry we're using. And then we have 2 different bioinformatics pipelines we're using to analyze the data. For the 18S samples, we use QIIME2 and the Silva 138 OTU dataset. And for the 16S and mute-S octocoral libraries, because their taxonomy is poorly understood in octocorals, we use a custom RAXML-based pipeline with a in-house developed reference voucher library set to do species assignments. So first I want to talk to you about this kind of initial exploration strategy. And what I'm going to be talking to you today is the results of a partnership between NOAA Ocean Exploration and NOAA Fisheries to test out the use of eDNA on the NOAA Ship Okeanos Explorer. This work was carried out in May of 2021 on a technology demonstration expedition and was a pilot project for assisting the program with using this new technique to collect more of a biological baseline of data for deep sea habitats. It's kind of a great partnership because ocean exploration has the technology to access these habitats, and NOAA Fisheries really has a lot of expertise in processing environmental DNA samples. So the work was carried out during the 2021 technology demonstration expedition. We conducted all our water sampling via CTD casts along the Blake Plateau, which is off the southeastern United States. We conducted 12 CTD casts, 8 in deep water and 4 in shallower water as other technology demonstration work was ongoing. Some of these casts targeted coral mounds, and in total we collected 120 environmental DNA samples. These samples were collected such that they were spaced throughout the water column on the upcast. So we collected samples on the seafloor, just above the seafloor, 25 meters up, so going up in 10-meter steps. We then made sure we targeted samples just below the deep scattering layer, within the deep scattering layer, and just above it, and then at the chlorophyll max and at the surface, spacing any additional available Niskin bottles throughout the rest of the water column. The goal being to really target being able to get that biological biodiversity snapshot of the whole water column so that we can see kind of not just what's on the seafloor, but what those food sources in the upper water column and in the deep scattering layer might be that may be then trickling down, and assess how these different water column habitats interact. So we took these back to the lab, have been working on sequencing through all of them, and I want to give you kind of a snapshot of what the initial results we're getting look like. So once we've run our data through a biological pipeline, we get out species assignments that we can sort in different ways to look at what's in each sample. And so this is what you're looking at here. So this is one CTD cast, and what you're going to see throughout this is going from left to right across this figure. Don't worry about reading these individual little tiny species assignments. Just know that each block of color corresponds to one type of taxa that we've been able to identify in the sample. And going from left to right across these different columns, what you're seeing is on the far left, the negative control, and then sample the deepest samples all the way up to the surface. So again, this is one CTD cast. And so we can examine the taxa present across our entire sample and sort and organize by what types of animals we're seeing. And so this figure you're looking at here is the taxonomic assignments from that CHIME2 pipeline. So let's dial in and really look at what we're seeing throughout the water column on the Blake Plateau. So one feature present in all of our samples is unclassified sequence. So this is sequence that doesn't match anything in the database, or what it matches is not a known organism. So these are largely unclassified eukaryotic microorganisms. They're present in the databases increasingly as more and more groups do this kind of environmental sequencing and have yet to be able to culture the individual organisms associated with these sequences. So because we can't get any more biological information out of them, we tend to remove these sequences from all of the analysis. So after we've done that, we can then dial back in and look at what we're seeing in each of our depths. So first I wanted to dial in and focus on the negative control, this furthest left bar. We were actually able to sequence DNA out of our negative control sample. Now the negative control was ship tap water from the wet lab on the ship, and we obtained high quality sequences from it. However, what's important to note is that none of these sequences overlapped with anything in the environmental samples. In fact, all the sequences we obtained were from nematodes, fungus, and amoebas. And so since this negative control is very, very different from anything we see in the environment, we can have high confidence that we're not getting any cross-contamination between our samples. And we ran one of these negative controls on each of our casts. This is also a good time to highlight why we call non-potable water on ships and labs non-potable. It's not necessarily treated the same way, and you should really, really not drink it. So looking at our environmental samples, we can start looking for patterns in the types of organisms we're seeing throughout the water column. So for instance, this group highlighted here in green is found primarily at the deep depths. So this is our deepest sample, just 5 meters off the seafloor, up to just below the deep scattering layer, and then it peters out as we get closer and closer to the surface. These sequences all come from the Rhizaria. So this is the taxonomic group that contains the foraminifera and radiolarians, which are a microorganism group that can be quite common in benthic sediments and also found throughout the water column. So it's good to get nice confirmation that this very common, well-known group that we know is deep is dominating our sequences from our deepest samples. As we move up through the water column, we can see that different other organisms start to make up a larger and larger proportion of the sequences we see. So if we look at this next group highlighted here in orange, we see that this taxonomic group has large peaks of sequence, which correspond approximately to higher, like a higher biological fraction within the sample. And these peak out within the deep scattering layer. And then this is the chlorophyll max layer. And when we look at what the taxonomic assignment of this is, these are copepods, and they're actually dominated by 2 different taxonomic groups of copepods between the deep scattering layer and this chlorophyll max layer. And actually these images you're seeing here are the 2 most dominant species, one from each group. So the Sapphirina opalina, sapphirinid copepods are most common within the deep scattering layer. And then this other species is found that has this long feathery tail, is very common within the chlorophyll max zone. And so going forward, we can also start to look at other taxonomic groups using additional markers. So in all of our deepest samples, we are detecting other benthic organisms, deep sea corals, ophiuroids. And the plan going forward with the rest of these samples is to look at that biological biodiversity, see how it changes throughout the water column, but then compare distributions going north-south in different environmental regimes. So do the patterns of organisms we see further south change as we get further north? Do we see different patterns of biodiversity from samples that were taken within the Gulf Stream versus outside of the Gulf Stream? And better characterize each of these different individual habitats. So we can also use environmental DNA to do slightly different types of exploration. This is more targeted exploration with specific management concerns in mind. And to give you an example of this, I want to talk about some work that was done in the 2018 and 2019 Express cruises on the West Coast. Express stands for Expanding Pacific Research and Exploration of Submerged Systems. This was a joint effort between NOAA, USGS, BOEM, and a few other groups, including MBARI. And the overall goal of the program was to enhance exploration of deep sea areas off the United States West Coast to inform management decisions regarding wise use of living marine resources, inform potential offshore wind energy and mineral resource decisions, and improve offshore earthquake, landslide, tsunami, and nautical hazard assessments. And so the nice thing about this work is it also was not dependent on a single ship or cruise. It used multiple vessels to do multi-agency work across multiple years. Now, one of the targets that I was particularly interested in was the exploration that was ongoing to explore deep sea coral and sponge habitats as part of the Deep Sea Coral Research and Technology Program's West Coast Research Initiative, which was one of the partners in Express. So our goal was to locate and characterize deep sea coral and sponge and chemosynthetic communities, including their associated fish populations. And we targeted coral and sponge habitats during the 2018 and 2019 cruises coast-wide. In particular, our focus from a management perspective was to do surveys that aligned with the proposed, at the time, Amendment 28 essential fish habitat boundary changes that have since been implemented. And so we were going in to do both visual and environmental DNA collection surveys across boundary lines that were going to be changing as of 2020, so that we would have baseline data in place so that we could go back after the changes happen in 10 years and see what impacts that changing essential fish habitat boundaries may have had in these zones. So we surveyed habitats from Oregon to California. 29 individual sites were surveyed across 2 years. We did visual surveys via both ROVs and NOAA's seabed AUV, and we also collected samples, both physical samples and environmental samples with the ROVs, and collected additional environmental samples, or eDNA samples with the CTD rosette on the ship. So I want to take you through some of the data we've gotten from this cruise effort, and I'm going to focus in on 2 different areas. The first is Daisy Bank. Daisy Bank has been the subject of a number of previous surveys. It was initially closed as an essential fish habitat area in 2006, and so this was the first chance to go back and survey it since that closure. So we completed 5 ROV dives on Daisy Bank. We surveyed a diverse variety of habitats on the bank, including sponge gardens, pavements and carbonate areas, and a methane seep. And we also collected numerous coral sponge and environmental DNA samples. We sequenced these samples for both corals and fish. And we were able to identify different coral communities on different parts of the bank. So for instance, in water collected alongside the methane seep, we were able to detect mushroom corals, a primnoid coral called Plumarella, as well as sea pens that are unknown to our current voucher collection. We also visually observed 2 species of coral, Swiftia simplex and Clavularia, which is a low-lying stoloniferous soft coral. However, we haven't detected those in our environmental DNA sequencing yet. And I want to highlight this because we can have differences between what we see in the eDNA and what we see in a visual survey, which is why they really complement each other well. They tend to overlap on a lot of species, and then each one can often detect additional species. In this case, we haven't sequenced this sample to the depth we would like quite yet, and we may get these remaining 2 species when we sequence the sample deeper, which is actually ongoing right now. We also looked at the rockfish community present at this methane seep, and were able to detect 6 or 7 different species, including species that were not observed in the visual survey. And we're using this data looking at the groundfish community and the deep sea coral community to better understand how groundfish are using these deep sea coral and methane seep habitats. The other really exciting site that we surveyed on the 2018 cruise is a site called Mendocino Ridge. This is the site of a new essential fish habitat zone, and this was the first visual survey that had been done with ROVs there. In this case, we took 3 eDNA samples and numerous physical samples. The picture you're looking at was taken at Mendocino Ridge, and it was very characteristic of the environment we saw there. There was really, really high coral diversity all over this site. It was very beautiful. And we took 1 CTD rosette sample and 2 eDNA samples via the ROV, which is not that many samples, and we're really excited to go back to the site in the future, given what we've seen even in this limited sampling panel. This hugely diverse area included a lot of species that we consider to be quote unquote northern, stuff that we see up into Alaska and British Columbia. It is not typically found this far south into California. Mendocino Ridge is in central California. These northern species included this white lace coral you're seeing in this image called Styraster columbiensis, as well as the habitat-forming coral Primnoa pacifica, which is also known as by the common name red tree coral, which is this very large, beautiful primnoid. And previous to this, had not really been observed this far south. It was thought to only extend to about Washington State. So from our CTD sample, we were able to detect a lot of the rockfish community present here, in addition to the coral community. And once again, we observed a lot of rockfish in the eDNA sample that we didn't necessarily see in the visual survey. We also were able to get coral DNA out of the CTD bottom water sample, which is really nice. It's again a confirmation that you don't necessarily have to have an ROV right in the habitat. If you can get a CTD rosette towards the bottom, within about 5 meters of the bottom, it's another really good way to take benthic habitat samples. So from our CTD sample, we found species including Primnoa, Characterize, which is a bamboo coral, some Plexaurid gorgonians, and a lot of mushroom corals. And I want to highlight this Characterize and Calcigorgia. The bamboo coral Characterize is not typically found at this depth range. These samples were taken between about 500 and 300 meters depth. And typically Characterize is found at 700 meters or deeper. And Calcigorgia is another one of these northern species that we typically don't observe south of about Washington State. So this just further reinforces that Mendocino is actually a really interesting, highly diverse zone that for some reason is providing optimal octocoral habitat for even species we don't typically see at these depths or at this geographic range. We also successfully were able to identify octocoral species out of the 2 eDNA samples we collected with the ROV. Again, we're seeing a lot of Primnoa, we see a lot of soft corals, we start seeing some of the bubblegum corals. And what I really want to highlight here is we also are seeing what I've been put here and highlighted unknown gorgonian. So this is the same sequence detected in 2 independent samples. And it's a gorgonian. It appears to be related distantly to the Acanthogorgidae clade, but it's not something we have in our current voucher collection. And getting it twice with pretty high sequence coverage is really good indication that there's a species here in this habitat that has yet to be collected on this coast. And that's actually an interesting point because eDNA collection can be iterative. We can use the results of an initial environmental DNA survey, not just to characterize the biodiversity, but also to identify potential future collection targets and surveys. So we're actually really hoping to get back to Mendocino Ridge on a survey that's coming up this fall. And we're going to be keeping our eye out for a new gorgonian that we haven't collected yet because we've been able to detect it in the environmental DNA. So overall, the Express cruise has also just been a really valuable source of new samples. We collected Chromoplexaura bankensis, which we collected the paratype, which is the second sample of a new species. That's a recently described gorgonian. It's this little yellow one. We were also able to collect a lot of Plexaurid gorgonians and soft corals that we knew were there. We have been imaging them for many years, but had not yet been able to collect to add to our DNA voucher collection. So we've now expanded our voucher collection so that in future environmental DNA surveys, our ability to detect and determine individual species will be enhanced. So we can use environmental DNA for exploration, but we can also use other omics technologies to explore the biology of individual species. And specifically what I'm going to talk a little bit about today is how once we have identified a species in a habitat, characterized its range, we can use omics tools to study the biology of the individual species, in this case, the population connectivity. So to do this, we use a technique called rad-tag sequencing. This is a technique that uses Illumina sequencing to sequence the region directly adjacent to what's called a restriction site in the genome. And basically what this is a way for us to reduce genomic complexity so that in a species like an octocoral, where we don't have a genome sequence available, we can still identify thousands of molecular markers that we can use across multiple individuals to characterize things like population boundaries. Each of the samples we run are barcoded, and this allows us to run hundreds of individuals at a time and separate them out later bioinformatically. And this whole process allows the rapid identification and genotyping of thousands of novel single nucleotide polymorphisms, or SNPs, in non-model species such as octocorals. And there's now a number of sub-techniques to this technique that improve marker reproducibility and can generate hundreds to thousands of additional markers. So why are we interested in population connectivity? Well, knowing how populations are connected can allow us to identify patterns of larval dispersal and identify individual populations at risk. Using omics tools like RAD sequencing, because we get thousands of markers, we can maximize the amount of data we get out of a limited numbers of samples. We don't need as many physical individual samples to be able to detect population boundaries when we're using thousands of markers. And to date, we've carried out RAD sequencing in 6 broad coral taxa. These range from Primnoa to several Plexaurid gorgonians to soft corals from Alaska, and some sea pens. So today I'm going to be focusing just on one of these groups, and that's Primnoa pacifica, specifically groups of Primnoa from Southeast Alaska. We're interested in knowing about population connectivity because different corals have different life histories in terms of how they spawn and disperse their larvae. And this means that they can have different susceptibility to anthropogenic stressors and different abilities to recover. And so when we're looking at population connectivity, we will be looking at it in multiple different types of corals because the connectivity patterns you see in one coral may not correspond to the connectivity patterns you see in a different species. So we've carried out RAD sequencing in Primnoa pacifica, which is, as I said earlier, the red tree coral. You're seeing an image of it here in the background. These are a long-lived and relatively slow-growing species. They can grow into colonies that can be several meters across, and that means that they're also very important as habitat for fish, including things like commercially important groundfish species. They have a patchy distribution, so you tend to find them in groves that are, you might find 10 to 20 individuals and then go for 50 to 100 kilometers before you see another one and then find another patch. But they are found across the Northeast Pacific from Central California all the way out to the end of the Aleutians chain. So they have a very broad overall distribution. And while we're only starting to really understand the reproductive cycles of Primnoa pacifica, we know that their sister taxa in the East Coast have very high local larval retention. So when they spawn, most of the larvae tend to settle around the adult colonies. And so we wanted to find out to what extent that is or is not true about Primnoa pacifica in the Northeast Pacific. So we carried out SNP development and RAD sequencing across Southeast Alaska. These individuals also were carried through a microsatellite study. So we have 2 types of data, SNP data and microsatellite data for these individuals. And we discovered more than 1,100 SNPs across 137 individuals spanning these 5 populations you see here. So we had 3 populations from the outer shelf and 2 that are from fjords in Southeast Alaska. We're still finalizing the final little bit of the analysis, but we have been able to detect some population structure across Southeast Alaska. So, and that's what you're seeing here. So the colors you're seeing on the map here correspond to each of these colors here. So what we've done is this is a principal coordinates analysis of about 800 SNPs across these 5 populations. And it's a little hard to see here, but what you can see is that this red population from Dixon Entrance, while it does have some overlap with the 2 Glacier Bay populations, does appear to be different than the 2 outer shelf populations we have from further north. And that also holds true in our structure analysis. This population number 1 right here does appear to be different than the other 3 populations here. And that's the only significant FST we find when we look among the different populations. And so for some reason, we do have population connectivity amongst these 4 northern populations and limited connectivity between this Dixon entrance population and these 2 populations up in the fjords, but very little genetic exchange between these outer shelf populations and this population at Dixon entrance. And we started to dig into why that might be, and what we found is that this pattern of connectivity actually matches what's known about the patterns of ocean circulation for this region in Alaska. You have this current that runs north for most of the year along the outer shelf, connecting these upper 2 populations. But right here at Dixon entrance, you can form a gyre and actually have water that's formed south. And so it appears that this gyre structure and some of the way the current change moves is keeping a lot of any of the gametes or larvae that are produced here from moving north into these more northern habitats. What's really interested and we want to explore going further is, since we know that some of this water through Hecate Strait gets transported south, we're interested now in looking at populations in British Columbia and Washington state to see if Dixon entrance here is a geographic break and whether individuals from Dixon might be more closely related to populations that are further south rather than populations that are further north. So we're currently actively collecting additional samples from British Columbia and the US West Coast to do a follow-up study to the study in Southeast Alaska and get a better idea of the overall patterns of connectivity in from NOAA Pacific and across the Northeast Pacific. And here I just want to highlight again why it's important to do this process in any species of interest, because the patterns we see are different among species. So in 2016, we carried out a rad study in a plexaurid gorgonian, a little sea fan called Swiftia simplex. And what we found, in contrast to what we're seeing in Southeast Alaska and from NOAA Pacifica, where we do have some population structure that we can detect, in Swiftia simplex, we had one panmictic population that went from Neah Bay, northern tip of Washington state, all the way to Southern California. And so if we're thinking about things like capacity for species recovery from disturbance, a big panmictic population is an indication that those are really common and they can grow quickly and are spreading, versus a population like Dixon that might be a little more isolated and might have a harder time recovering from disturbance. And so we have to look at these kind of connectivity patterns and larval dispersal patterns in individual species, because connectivity can be variable. So just to summarize, genetic and genomic tools can expand access to hard to reach habitats and make the most of limited samples. We can apply high throughput sequencing to taxa to better understand connectivity and cryptic speciation, which is important for conservation and management. And novel techniques such as eDNA sequencing can provide detailed biodiversity data beyond traditional sampling and surveys. So I'd like to thank the team that supports me at Northwest Fisheries Science Center at the lab there, as well as all of these different participants who've been really key to collecting these samples, doing the field work, supporting the research in the lab. It wouldn't be possible without a lot of partnerships. And with that, I am happy to take any questions.
Excellent. Thank you so much, Meredith. That was a fantastic presentation. I just want to say how fun it was to get an opportunity to stay with you last year and test out the use of eDNA for the first time in NOAA's Office of Ocean Exploration and Research. So awesome to see those results and all of the other projects you are working on. So folks, there's lots of questions coming in, which is great. So feel free to type your questions into the questions for staff box. So Meredith, I'm going to start reading these out loud for you. Our first question is from Damien. How do you account for ocean currents in relation to where the samples were collected versus where the DNA originated?
So that is actually one of the, as I would put it, million-dollar questions that I'm working on right now. So in running the deep sea coral samples that we've run from the West Coast and the Central Pacific, what we see is if we can see a high current in the ROV, often, even if we're sitting right next to a big coral colony, we won't be able to detect the eDNA, which is a good indication that, yeah, as you would expect, with high current, the water mass you're measuring is not the water mass that is right there at the animal. So whenever I can these days, if the ship I'm operating on has an ADCP, I try to get an ADCP current reading before I take the environmental DNA sample. And in the work we're doing from the express samples, one of the things we're looking at is correlating what that ADCP looks like, current direction, where it was taken, with the environmental DNA samples, and just seeing how that drives the different patterns. So that is an area of ongoing focus right now.
Excellent. Thank you so much, Meredith. Our next question comes from Tom. There are often population differences in corals between different depths. This did not seem to be the case with P. pacifica.
No, we actually, we are looking at isolation by depth, and we have not seen it in Primnoa pacifica. In the other species we're looking at it again. We have a sea pen species called Helipterus, where it actually looks like we have a taxonomic shift in that genus that occurs with depth. And in some of our Plexaurid gorgonian populations that we just now are analyzing data for, we're also going to be looking for isolation by depth. It's been really well documented in the Gulf of Mexico and off the East Coast. So I do expect we're going to see isolation by depth in some of the species we see out here. We just are not seeing it in Primnoa for some reason.
All right, thanks, Meredith. The questions keep coming in, so we have a couple more here. This next one is from Paul, who starts off by saying, thanks, Meredith, so much cool stuff here. Did you recover tunicate sequences? Are there many deep sea tunicate species?
There are quite a few deep sea tunicate species. I have not taken a deep dive into the data we've gotten with the 18S yet to look for tunicates specifically, but in the benthic samples, my guess is if I pull up that data, we will see them.
Great, thank you. Our next question is from Danielle, who also says, thanks for the presentation and interesting to see the results of your work. She asks, where was the sample of species name, C. cordelbankensis collected?
Chromoplexara cordelbankensis. So we actually on that cruise collected two, one from just north of the Channel Islands and one from within the Channel Islands. And both of those specimens are currently housed at Cal Academy, so they're available for other researchers.
Excellent, thank you. We have a question that came in from Chris, who's asking if there's an online site to access photos. I'm happy to take this question. Chris, I'll drop some links here in the chat, and for the rest of our audience as well, can access photos from NOAA Ocean Exploration, and the Deep Sea Coral Research and Technology Program at NOAA also has photos on their website. So I'll get to that.
Yeah, and to give a plug for Deep Sea Coral, they are currently working on revising the website to include even more photos and link to the tools that Ocean Exploration has as well, including species ID guides.
Great news. Okay, thanks, Meredith. Our next question comes from Paul. You described limited divergence among Primnoa locations. Do deep sea coral gametes disperse at depth? How do ocean currents at depth differ from surface transport we're more familiar with?
So this can get really complicated with deep sea corals. Corals in general have two life history strategies. They can be what we call broadcast spawners, where they will broadcast their gametes up into the water column, where depending on what local currents are, and those can vary depending on where you are in the deep sea and how high up or low in the water column those gametes go. But they basically go up into the water column where they combine and then get carried to wherever they're going and settle out. But deep sea corals can also have what they call a brooding strategy, where they basically only release one or the other gamete and then brood the larvae within their own tissue, and then a fully formed little mini polyp will pop out from the adult and basically settle right next to it. And so broadcast spawning corals can actually have really wide connectivity like we see in Swiftia simplex. That's a known broadcast spawner. And yeah, so those gametes go everywhere. We've got one panmictic population up and down the coast. With Primnoa pacifica, they are a broadcast spawner, and there is definitely some connectivity. So in some population, we definitely are seeing gamete transport. No one has actually collected like deep sea coral larvae out of the water column, so we don't really know what that looks like. We have to detect it genetically. But other Primnoa species like Primnoa rosetiformis can have really, really high larval retention. So even though they broadcast spawn, they're often in canyons, and the larvae just don't go that far. So it can be really complicated just depending on what the local oceanography is.
Great, thanks so much, Meredith. We have a comment here from Susan who says, fantastic presentation. Thank you. I'm seeing terrific application of this information for future middle school and high school education. I completely agree, Susan. I don't know if you just wanted to quickly comment on that, Meredith.
Yeah, it's great to see this stuff get used. I know both NOAA Ocean Exploration and then through the Deep Sea Coral Research and Technology Program and the Ocean Exploration Trust, all three of these groups have really great classroom materials that are available and curricula for classrooms. So explore through those. They've got great resources, including images and lesson plans that you can present to classes of various ages.
Thanks, Meredith. Okay, we have a question from Paul. Mendo is a significant transition / contact zone for many species. What are current thoughts about the biogeography of this interesting feature?
We actually, so when we're going into doing the survey at Mendo, usually we think of Point Arena further south as being the major biogeographic boundary for a lot of our coral species on the West Coast, but it's looking like, yeah, Mendo may be one. And so we've only been able to do one ROV survey there, and we're really hoping to go back this fall and get a few more dives in there to really better understand how this site acts as a geographic break. So stay tuned.
Great, thank you. Okay, our next question is from Francisco. I may have missed this, but does collecting eDNA include ocean floor sampling with ocean water samples?
Yes, so we are, when we're doing eDNA for benthic, the benthic surveys, we're using Niskin bottles either on the CTD rosette or mounted on an ROV vehicle to collect bottom water samples from the habitat of interest. I hope that answers the question.
All right, our next question is from Severin. Thanks for the great presentation. My question is whether the mute-S marker alone is sufficient for species identification of octocorals. Molecular analyses of octocorals still seem to be quite difficult, and mostly the 28S marker is used additionally.
Yes, so this is it's an ongoing focus of research, both in our lab and then a lot of work being done at the Smithsonian and Lehigh University. So we're using mute-S because it does a fairly good job, but an imperfect job at distinguishing species. I'm currently looking into using 28S. What I have found in the species I've run it on so far is that much like mute-S, it does really well at some groups, but not others. But I'm doing more work developing it. Eventually, we would like to do multi-marker panels for the eDNA work, doing both 28S and mute-S, because the more markers you get, the better your species, the better you can distinguish individual species within these samples. And so we are looking into starting to apply 28S. We are going to be sequencing our voucher collection for that as we move forward, in addition to the mute-S marker. So yeah, octocorals continue to be kind of a taxonomic puzzle, and every time we think we have them dialed in, they throw us another loop. So.
Great, thanks, Meredith. We have just a couple of questions left. This one is from Mary. Can you detect associated invertebrates with corals?
Yes, so we can't use the mute S marker because it's octocoral specific, but if we include a CO1 marker or an 18S, we can detect associated invertebrate communities as well. We haven't done that with the West Coast work because the interest from a management perspective has been the fish communities associated. But for some upcoming work in the Central Pacific, we're looking into looking at the associated invertebrate communities as well.
All right, great. Thanks, Meredith. Our last question is from Francisco. Is there a time of the year where ocean currents is minimal for collecting data for eDNA?
That's going to be really region specific. So as you saw in that slide from Alaska, just seasonally, you can have a complete shift in direction in current. And so you're going to need to know, get a highly detailed picture of the local oceanography at whatever site you're going to collect. So if you want to minimize current, you can look at things like tides, but you're really going to have to know the oceanography of a specific zone. And even then, the ocean's a big place, and you can have things like internal waves that can propagate unpredicted currents. So sometimes you just have to get lucky.
Excellent. Thanks so much, Meredith. I think that's the end of our questions. We did get a couple of comments about whether the presentation will be made available. Yes, we record all of our presentations and make them available up on our NOAA Omics website. So that is linked in the chat. Additionally, I provided links to NOAA Ocean Explorations and NOAA's Deep Sea Coral Research and Technology Program websites for folks to learn more about deep sea corals and ocean exploration in general. So with that, Meredith, just want to thank you so much again for presenting today, and I just want to thank everybody for joining us today. Just a reminder that the NOAA Omics Seminar Series takes place on the third Wednesday of every month at 12 p.m. Eastern Time. Our next seminar will be on June 15th, 2022 at 12 p.m. Eastern Time. And we have a speaker from Alaska's Fisheries Science Center who will be giving a presentation that day. So with that, I will go ahead and close out the seminar. Thanks, everyone.